Hello, YouTube, and welcome back to Science with Sully. If you like the video, be sure to give me a follow and a like. And if you really like the video and want more content, be sure to subscribe to the channel. Today, we're going to be talking about two processes that kind of go hand in hand together. But we're going to talk about pinocytosis first, and then we're going to talk about phagocytosis after. Both of these kind of go hand in hand. The mechanisms are virtually the exact same, but they are different processes, so we need to talk about them separately. So first, we will talk about pinocytosis. Pinocytosis, in general, you are bringing stuff into the cell. And if you remember in my last video, I talked about endocytosis and how it was usually very specific. Pinocytosis is not. It's usually not specific. And one other thing that I like to do is to break down the word that we are trying to, the mechanism that we're trying to learn. I like to break down the prefix and suffix. So pino. Pino is a Greek word and it means to drink. And cytosis, as you probably know, just refers to the cell. So it's the cell and it's drinking extracellular fluid and bringing that substance in. So let's get to the mechanism. First, we want to draw our cell membrane, and it's a bilayer, as always. And this would be the outside of the cell, and this would be the inside. So our cells need to be able to bring in certain things, substances, molecules, liquids. The cells are mostly made of water, and sometimes they need to bring in water just to stay, to keep their structural integrity. Sometimes they need to bring in other things like proteins or large bulky items. So let's draw a couple of things that we're going to want. We'll want these stars, and those will be a substance, and these squares will be a substance. Let's draw a couple stars over here. So these are our substances. We don't really care what they are. We just know that we want these substances to go into the cell. So basically, our cells can sense when these substances are out here through a couple of different mechanisms. But usually there's a protein, a plasma membrane protein that will be stimulated by one of these, and that will kick off the process of pinocytosis. What the protein is not as much important in this video as the big picture of pinocytosis. And what the cell is going to do, it is literally going to drink some of this extracellular matrix and just bring it in form of a vesicle. So how's this going to happen? Let's take a look. So the cell realizes that it wants both of these substances in there. So what it's going to do is it's going to create a pit in its membrane like so. Similar to endocytosis, but it's not as specific as endocytosis. We want to bring in the stars and the squares. So we want both of these to go in. Well, when this invagination or this dip in the cell membrane happens, it pulls the substances with the water that they're floating in into the pit. What happens? We have proteins on the cell membrane that will push these sides of the membranes together and fuse them like so, with our substances in the pit. So it's drinking all of this matrix, water and all. So what happens next? Well, once these two sides come together and fuse, you have a vesicle. And in that vesicle, you have got your substances that you wanted to bring in. And the vesicles are also bilayered, just like the membrane. And then these vesicles can travel along our filamentous networks in the cell, and both can go to their destination, the substances, and cause intracellular changes or nutrients that the cell needs. So this is pinocytosis. Remember, the big takeaway of pinocytosis is it is not specific. It is grabbing everything that's out there. So even if it wanted just the blue squares, it's still going to get a couple of the red stars. So that's pinocytosis. So now let's move to phagocytosis. Phagocytosis, the mechanism is actually the exact same. So we will first take a couple of notes about it and then look at the mechanism and look at the differences in the mechanisms of phagocytosis and pinocytosis. Phagocytosis is the consumption of cells, or we can say the consumption of antigens / pathogens. So that should be ringing a bell. Ding, ding, ding. Those two words. Phagocytosis is a process in immunity, or the immune system. So what type of cells practice phagocytosis? They're macrophages. And these are immune system cells. So they are also immunity. Let's break down the word phagocytosis, and then we'll break down the word macrophage, and then we'll get into the mechanism. So, phagocytosis. Phago is Latin for the phrase to devour. Easy to think. We are consuming something, and it's usually a pathogen or antigen that we are consuming. And as we've discussed before, cytosis just refers to the cell. And let's go ahead and break down the word macrophage. Macro is just Latin. You've probably heard that prefix for large. And phage, phago is the verb to devour. Phage would be the noun, so it is the devourer. So a macrophage is a large devourer. And you will see that in a moment. Let's get into the mechanism. So we're going to have our cell membrane bilayer always. And this would be the outside, or the extracellular matrix. This would be the inside, or the cytosol. So I discussed, if this is phagocytosis we're talking about, the type of cell that we are looking at, that we are zoomed in on, is a macrophage, because they're the cells that do this process. So what happens? Well, whenever we get sick, in our extracellular matrix, in this example, we'll be using bacteria. So we have a bacteria that's floating around in our extracellular matrix. Let's put some lipopolysaccharides on there, make them all nasty and sick looking. So that is a bacteria. And that would be our pathogen. So that's what's going to get consumed by our macrophage. So what's going to happen? Well, the macrophage has receptors on it. Let's see. The macrophage will have a receptor on it that will fit these lipopolysaccharides in it and stimulate the cell to undergo phagocytosis. And these lipopolysaccharides on the side that stimulate this, this receptor are called epitopes. Just for a little random fact. But in a gram-negative cell, they would be lipopolysaccharides. So they're stimulating this macrophage, and this macrophage is going to make some intracellular changes downstream that are going to stimulate phagocytosis of this bacterium. So once this happens, we'll take this to the side, and you'll see something similar to our other cytosis, endocytosis, and really closer to pinocytosis videos. But what happens is this downstream effect is going to promote phagocytosis, which is what we're looking at right here, and a large, really, really large invagination of the, or pit will be formed, and that will in turn bring in our bacterium or our pathogen. It could also be a virus. And now it is sitting nice and snug in our pit. And as we've seen many times before, we have proteins on the plasma membrane that are going to pull towards these sides towards each other's, and they will fuse. So right there will be membrane fusion. And when those two sides fuse, you could probably see it's going to form a vesicle. And that vesicle will contain our pathogen. And this vesicle is a special type of vesicle because it's in a macrophage. We would call this a phagosome. But it will end up acting a lot like a lysosome, and it will lower the pH and increase a bunch of hydrolases, and it's going to have a bunch of proteases, and all of these things together are going to break down our pathogen into nothing. It's going to kill it and protect us from getting the effects of that pathogen. So this will end up a regular, just an empty vesicle eventually, and that vesicle will go back and fuse to the membrane and do it all again next time a pathogen is in the extracellular space or matrix. So that's a quick rundown of phagocytosis. The big takeaways are phagocytosis, and I didn't really say before, this is specific. I know it's the same type of kind of mechanism as pinocytosis, but what the key difference is we have these epitopes on the bacteria that we're trying to consume or devour, as what our prefix means. And we're going to bring that in, break down the bacterium, Then we're going to have our vesicle recycled and sent back to the plasma membrane. If you like this video, be sure to give the video a like and the channel a follow or subscription. And thank you for joining Science with Sully. And I will see you next time.